Understanding vision, approach, and motivation. In vision, visual inputs to the retina are first image processed and then sent to the primary visual cortex, called V1, the first cortical area to receive retinal signals. More than half a century ago, retinal neurons are discovered to respond to dots in visual images, such as a white dot in a black background or a black dot in a white background, a red dot, a yellow dot, etc. Different neurons respond to different dots, and the locations of these dots tie up the whole visual field. Soon after, Hubel and Wiesel discovered that V1 neurons are excited by short lines or edges. One could use similar approaches to study higher visual cortical areas around the visual pathway, like V2, V3, V4, etc., and explore how images of more complex objects such as faces, houses, trees could excite those neurons. However, decades later, we have not gained as much insight as perhaps expected by Hubel and Wiesel. Let's try to push the frontier of research beyond V1 by asking new questions. So we could ask, what is vision? To frame vision as having a low level, a mid-level, and a high level visual stages is not precise enough to guide us and is difficult to falsify. A more precise framework was by David Marr a few decades ago, posing vision as made of these three stages. Make a primal sketch, then 2.5D models, and then 3D models of the visual world. However, progress by this framework has been very limited. Now, the new proposal is that vision has these three stages: encoding, selection, and decoding. Only a very small fraction of visual input information is selected into the attentional bottleneck before being recognized or decoded, and this framework puts selection in a center stage. Critically, this selection starts at V1's output to downstream areas, and this means we need to examine and understand V1 much more than Hubel and Wiesel's discovery. The downstream visual cortical areas and the next stage of visual processing have to be understood in light of this information bottleneck, which implies that we are blind to most of the visual inputs to the retina. In this framework, selection and decoding are like looking and seeing. We will see how looking starts by a saliency map in V1 to guide eye movements, and how this framework enables fresh progress beyond V1. Using theoretical methods and working closely with experiments, this framework is easily tested and falsifiable by experiments. Let's examine the idea, the progress, and where they lead us to.